The molecular principles underlying DNA replication have been harnessed for major breakthroughs in biotechnology and medicine. One of the best examples is the polymerase chain reaction, or PCR, a technique that directly mimics natural DNA replication. In PCR, during denaturation, heat replaces helicase to separate the DNA strands. In annealing, short synthetic primers bind to the template strands just as primates-produced primers do in cells. Then during extension, a thermostable DNA polymerase builds new strands by adding nucleotides to primers, just like DNA polymerase inside living cells. Understanding replication has also transformed medicine. For example, quinolone antibiotics block bacterial topoisomerases such as DNA gyrase, which differ from human enzymes. Normally, gyrase cuts and reseals DNA to relieve supercoiling, but when quinolones like ciprofloxacin bind, they trap the enzyme with DNA still broken, halting replication and killing the bacterium. Acyclovir, used against herpes viruses in infected human cells, once activated by a viral enzyme called thymidine kinase, is incorporated into viral DNA where it blocks further replication, stopping the virus from making copies of itself. But because it requires activation by a viral enzyme, it spares healthy human cells. Replication principles also power anticancer therapy. Because cancer cells divide much faster than normal cells, they need a constant supply of nucleotides and active DNA synthesis. This makes them especially vulnerable to drugs that interfere with DNA replication. Methotrexate blocks the production of DNA building blocks. 5-fluorouracil mimics natural nucleotides and disrupts polymerase activity. Topoisomerase inhibitors such as doxorubicin and etoposide trap enzyme DNA complexes, causing double-stranded breaks that lead to cell death. So understanding DNA replication is a driver of innovation in biotechnology and medicine.